Hello, my name is Ellie, and I'm going to be covering the methods section of our research project. All right, so I broke it down into 3 different sections. Step 1 is sample collection, step 2 is DNA extraction, and step 3 is DNA results. So step 1, sample collection, is arguably the most crucial step in this entire process, as poor sample collection will affect everything downstream. So the first step to obtaining a species identification or a successful species identification is sterile plant collection techniques with an emphasis on sterile. For any plant evidence found in a crime scene that is to be collected, samples should be stored separately, even if they look similar. And this protocol is in place because most forensic crime scene techs and law enforcement officers are not botanists and may mistake 2 similar looking plant parts as being from the same plant. Tools should not be reused when collecting plant samples, so technicians should have single-use tongs, gloves, and bags on hand, or anything needed to collect that plant sample. If they do not separate out their tools or mix this 2 different plants into a bag, this will lead to a mixed genome result and will not yield usable data. The second crucial step is proper storage to prevent molding and further degradation, which could lead to the loss of DNA. While degrading plant matter can still be run through the extraction process, it does run a higher risk of having poorer outcome, as it will have damaged and degraded DNA. To ensure the plant will not degrade further, it is best to either expose the sample to liquid nitrogen or store them with gel silica packs, which is more feasible. This ensures that the plant dries fully within a 12-hour time window and will remain stable for testing. So once all of that has been achieved, you can move on to step 2, which is DNA extraction. And everything for this section was pulled from Qiagen. They have a very competitive research-backed product. They actually have 3. And I do talk about the DNeasy Plant Mini Kit. The Pro Kit does have the best success rate, but the Plant Mini Kit is still very competitive compared to all other market kits. And this picture right here is essentially all DNA extractions for every kit, basically. So the first step the process of DNA extraction is step intensive, not only in terms of testing performed, but also to obtain the proper supernatant for said testing. So the Qiagen DNeasy Plant Mini Kit is a commercially available kit that has been verified for forensic use and has several studies backing its range of use. What's wonderful about this kit is that it only needs 0.5 to 1 whole gram of the sampled plant for the entire DNA extraction process, which is a very small amount. So the Qiagen breaks the extraction process down into 6 steps. The first is placing the 0.5 to 1 full gram of sample into a tube that contains a tissue disruptor, which is marked as CD1, as well as a bead beading step that is included in that. And you can't really tell in the picture, but it's like a tiny little, almost UFO looking bead at the very bottom, and it is typically metal. This will break down the tough plant cell walls to make access to the DNA easier for the later steps. The tubes will then go through a vortexer. After the step is completed, the supernatant will then be collected and transferred to a clean tube along with C2 for PCR inhibitor removal. Examples of a PCR inhibitor include polysaccharides, polyphenolics, and metabolites that came from the previous isolation steps. These tubes will be centrifuged and the supernatant collected, but it should be noted that disturbing the pellet should be avoided. The 4th step requires the supernatant to be transferred to a tube with a filter, which will be spun down several times to filter the supernatant. The supernatant will go through the filter, which will collect the DNA. Once it has been spun down several times, the filter column is moved to a clean tube. In all of these steps, you have to go through clean tubes. It will go to a clean tube and it will be washed several times with a buffer. The buffer is removed once the spin is complete, and a new buffer is used to wash for the next cycle. Once this step is completed, the filter column is moved to a clean tube and washed with a new buffer EB, and the column is discarded. The wash is now ready for PCR. So that is step 3. So the final step in obtaining a successful species identification is to run the extracted DNA through PCR and gel electrophoresis. The wash from the last step of the extraction process is added to a PCR master mix, which is usually trademarked by all companies, but they generally involve the same ingredients, maybe just slightly different ratios. and it is placed in the machine. For these types of samples, the thermocycler should be preset to denature at 94°C for 30 seconds, then annealing at 55 for 30 seconds, and extension at 72 for 60, and this should be set to repeat for 32 rounds. The final round of extension is 72 for 10 minutes. The denaturing step works to separate the two sides of the DNA strands to expose their nitrogenous bases, which are A, G, C, and T. Once these are exposed, we will cool down to the annealing phase. And this is where the master mix really comes into play here, because it contains nitrogenous bases and primers for that annealing and extension phase. So for the annealing phase, once the DNA has opened up and been exposed, the primer can come along and bind to the beginning part of a coding region of DNA. Once the primer has bound, we move to the extension phase, which is where we add our bases to create a new complementary strand of DNA. The steps repeat and we get exponential growth, which eventually ends and we achieve a sigmoidal curve of growth of PCR products. So you will begin a slow increase and then you basically almost go like straight up, and you essentially, you can take a really, really, really small amount of DNA and get a very high amount of DNA at the very end. So once the PCR process is complete, its products are taken on to an agarose gel and run through gel electrophoresis, which is the bottom picture under step 3. The banding created in the gel determines if the sample goes on to be sequenced, and one band is desired in this step, and every single identified species has one band in this picture. So once you achieve a single band, the sample is run through sequencing using the primer sequences for locus PSVA, TR and H, and TR and L, TR and F. And these locus were chosen after vigorous testing was done on plant locusts, which had the right variability to allow species identification. The discovered sequences are then pushed through the BLASTIN algorithm for a species match, and that is just an online database where you can go in, and it's actually really neat. You can type in really anything that can get you to a species match, whether it be the sequence that you obtained here. There's a bunch of other options on that website. It's very cool. But yes, this is how you get from pulling a plant in the field all the way down to obtaining your sequence, pushing it through a database and obtaining a result from that.